Aerpio Therapeutics is a privately held biotech company advancing first-in-class ophthalmology products. We have a small molecule Tie2 activator, which has completed a phase 2 study, and we also have an antibody that activates Tie2 as well. So this is actually a pipeline of products that will treat the various retinopathies. So Tie2 is a mechanism that is now broadly known in the literature to be very much in play in all the retinopathies, in wet AMD, in DME, in diabetic retinopathy, and also in RVO. Our approach to activating Tie2 is unique, and I'll go through the mechanism in a minute, because the mechanism is important here. And we have a portfolio of products that will give us proprietary products out through 2034, and we control all the IP on our programs. So again, the mechanism is important. So Tie2 is a transmembrane tyrosine kinase receptor. It's located on all vascular endothelial cells. And there's a whole plethora of literature, which we've benefited from, that substantiates it as being the key mechanism that stabilizes vessels, it blocks leak in vessels, and it also blocks pathologic angiogenesis. All things that are very important in diabetic retinopathy and DME in particular. Now, just to go over this in a little more detail, there's 3 factors that really regulate the Tie2 receptor. One is the activating ligand, angiopoietin-1. The other is an inactivating ligand, angiopoietin-2. But what everybody has ignored is the phosphatase, the vascular endothelial cell protein tyrosine phosphatase, otherwise known as VE-PTP. And it is the most downstream regulator of Tie2. So I think you ignore the phosphatase at your own peril. So what we did is we developed a small molecule, 9778, that blocks the intracellular domain of the phosphatase and prevents it from inactivating Tie2. And we have published literature that demonstrates that we can activate Tie2 both in cells and in animals. And to our knowledge, we're the only group that has published data that shows we can activate Tie2 in cells and in animals. Now, you may recall we presented our Time 2 data in previous meetings, and we've usually focused on the DME endpoint, which was central subfield thickness. But what we're most excited about from this trial is that we did an analysis, a pre-specified secondary analysis of the diabetic retinopathy severity score. And we were able to do this in both the study eye and the treatment eye. And the reason we can do that is our drug is dosed systemically, subcutaneously every day. And this trial was randomized into 3 groups. One was the monotherapy arm for 9778. The other was the combination arm with Lucentis, and then we had a Lucentis control. And so for the DRSS analysis in the study eye, we went according to the treatment groups, but in the fellow eye, we were able to pool the 9778 treated groups and compare them to the Lucentis treated patients, but the Lucentis treated patients didn't get Lucentis in the fellow eye, so it's actually a true placebo in that eye. And what was interesting is we got similar results in both the fellow eye and the study eye. We were able to get about a 10% improvement in 2-step improvers in the monotherapy arm, in Lucentis alone, as well as in the combination arm. Those are not statistically significantly different from each other. And we were able to duplicate that in the fellow eye, where 9778 was again able to give just north of a 10% improvement in the diabetic retinopathy severity score. So we're very excited about this result, and we're gearing up to do a longer study and a larger study to validate this in a 1-year diabetic retinopathy trial, treating patients with non-proliferative diabetic retinopathy. Now, we're not alone on focusing on diabetic retinopathy. Regeneron is now initiating the Panorama trial looking at EYLEA on longer interval dosing in non-proliferative diabetic retinopathy patients, and the DCRC net is also initiating trials in this space. So, however, we have an important advantage. 9778 is dosed systemically, so we treat both eyes, and we reduce the burden of the intravitreal injections. So I was pleased to follow Alimera, where they describe the burden of the intravitreal injections quite nicely. So we do think this will be a much more patient-friendly approach. Diabetics are very used to doing subcutaneous injections, and we can give our drug via 27, 28 gauge needle, similar to a insulin injection. Again, I want to focus on our mechanism and our pipeline. The antibody we have which also activates Tie2, binds to the extracellular domain of the phosphatase. And so this also replicates all the pharmacology we see with the small molecule. Now this brings along a drug that we can use for the more serious retinopathies like DME and wet AMD. And we can also do co-formulations of this with anti-VEGFs. So we think this will be a very powerful next generation drug for us, which will go after the more traditional approaches to treating diabetic macular edema as well as wet AMD. Again, we're not alone here. Roche and Regeneron have anti-angiopoietin approaches to trying to activate Tie2. However, it's not clear to us that the anti-angiopoietin 2 approach is going to activate Tie2 in as robust a way as we do. So the devil is in the detail, and the mechanism is important. So just to show you the Ang2 approaches, as shown on this slide, bind the Ang2 inhibitory ligand. However, it's unclear to us whether that's going to be sufficient to activate Tie2, because you need Ang1 around in order to activate Tie2. And in diabetic vasculature, you've stripped away the parasites, which produce Ang1, so there's not really a source. And you've also ignored the phosphatase, which is the most downstream regulator of Tie2. So we think our approach is likely to be the most robust approach to activating Tie2 and having a profound effect in these patients. Again, the antibody binds to that extracellular domain. It inhibits its interaction with the tyrosine kinase, and we do think that'll be a robust approach and allow us to do co-formulations with anti-VEGFs to develop products here. So we do expect this to work. We have tested our antibody intravitreally in animal models, and we're anxious to get it in the clinic, which we will be doing over the next year. So thank you for your attention, and thank you again for the invite, and we look forward to coming back in future years and presenting data.